not only about what motivates me to do this work, and you already understand what the biological carbon pump is about, so I'm not going to spend much time about it. So the big questions about the biological carbon pump regard the fact that it's an extremely dynamic and fast system. And so the question is, how does it move carbon from the surface into the deep sea? Is it stable? Is it predictable? How will it change? And how do we link satellite observations to flux or particle concentration? And Yun Hao and myself, Phoebe and Dan, wrote a paper, and that's what got the invite. The subtext of this, I'm going to get rid of this OK message. And maybe I can move this thing out of the way. Yes, okay. So I just used up 10% of my time. All right, I just want to thank a lot of people. They're peoples whose data I've referred to in this talk by Hannah Bourne. She wrote a PhD dissertation on particle dynamics in the California current system. Todd Wood, who's worked with me as in technical and engineering support. Beth Connors, who's done work on the Palmer Peninsula and just graduated from Scripps. Yun Hao Lee, who just is at Scripps. And Alejandro Morales, undergraduates, and of course the National Science Foundation has funded my research. Multiple people at Scripps, Wet Labs, now owned by Seabird, and MRV Systems, which is a float manufacturer, have contributed. I can't tell you how many ships I've been on. It's about 40. And I'll get to that later. And more important, we're concerned about how do we observe biological processes in dynamic ocean systems, and the only way to do it is through autonomous observations. So you'll see links to the California Current Long-Time Ecological Research Program, and also geotraces, which is mainly what the paper I'm talking about is. So this is a block diagram of the biological carbon pump, and most importantly, it does set the CO2 chemistry. And every time I edit this slide to show it, numbers up here in the atmosphere have gone up. And of course, we're concerned about acidification and the effects and how changes in ocean stratification, et cetera, affect how the biological carbon pump works and how it exports both particulate organic carbon and particulate inorganic carbon. So that's been my focus. And for my entire life, I've been interested in the chemistry of particulates. So in light of observations needed, I've collaborated with Scripps in the development of a device called the Carbon Explorer. It's a transdusometer version of an Argo float, and we've deployed them. And I'm going to come back to the Southern Ocean iron fertilization experiment at the end because there's some interesting messages. Anyway, this profiling float can carry sensors for particulate organic carbon and particulate inorganic carbon, and we've actually deployed them with those sensors. The Carbon Flux Explorer is a device to measure particle fluxes through imaging, and it used much of the same methodology as transmissometry and the PIC sensor, and most recently, we've developed a system called the Ocean Carbon Observer, which melds the capabilities of these two float systems. So we're motivated to develop this thing. I'm going to describe it very briefly and go on to talk about geotraces. So most importantly, in development of this new float, we've actually taught it how to measure particle size distributions and enabled a classification scheme using data published by Hannah Bourne as part of her PhD dissertation. And the main topic will be the analysis of satellite in-situ optical proxies and the Southern Ocean Surprise. So how did I get here? I've been all over the place. This is both the Atlantic and the Pacific, and you can add up all my time at sea. It's almost 2 years, 40 days of wire time. That's hardly a way of studying biological processes. But most importantly, particle sampling using the multiple unit large volume in situ filtration, the device that filters thousands of liters, combined with particle optics, led to the development and proof of the Carbon Explorer. And of course, the Carbon Flux Explorer basically has been developed and tested in the California current regime. So yes, if you go back to results or time of when I was in Canada at the University of Victoria, I went to sea multiple times with the large volume filtration system and collected particles at Station Papa. And sure enough, if you go and do a seasonal study, particulate and organic carbon is highly variable in space and time, and it just, there's no way to know how the system got from one state to the other without continuous observations. The further thing is particulate matter concentrations have seasonality that can be seen to kilometer depths. So that means that local observations with profiling optical sensors can detect things that bulk nutrient distributions can't see. This is the Carbon Explorer. The most important thing is that we deployed a transmissometer, and beam attenuation coefficient, or CP, is a function of particulate organic carbon concentration. And multiple places, this relationship is held up. The birefringent sensor that I'll be talking about has a laser. It looks just like a transmissometer. It has a laser. It shines laser light itself is polarized, and when that polarized light goes through a polarizer that cleans up aligned with that laser, then the beam becomes highly polarized. And when it shoots down the path length of the instrument and hits the detector, there's a polarizer rotated at 90 degrees. And so basically what you're seeing is black, no signal unless there are calcium carbonate particles in the beam. So the idea is you put a calcium carbonate particle in the beam, you can get, it's highly birefringent, which means that it has two refractive indices, which means that when laser light enters the particle, its plane of polarization gets altered, and the photons exiting the particle are rotated in a way that they can pass the detector. That's how this sensor works. We know it's unique. These are data from the Santa Barbara Basin showing in green just raw transmission voltages, in gray data from a scattering sensor, and in blue the up and down traces from this PIC sensor. And of course there are spikes in the data. We can filter those data, and here is the scattering data and superimpose on oxygen. And at the bottom of the Santa Barbara Basin, there's actually denitrification going on below 450 meters, and oxygen in the deepest layer is less than 2 micromolar. You can superimpose that beam attenuation coefficient, and basically the lines line up. In the deep water, they diverge from one another in the surface because scattering sensors, and beam attenuation coefficient are different inherent optical properties. And then we can show the PIC sensor, and the most important thing is that the systematics on the water column mirror the other particles sensors until you get below a depth of about 450 meters, and then it basically, we see that calcium carbonate has been dissolved. So we've repeated this observation at least 3 times in the Santa Barbara Basin, so we knew that this sensor is a unique inherent optical property. So what does a Carbon Flux Explorer do? This is the basis for the Ocean Carbon Observer. It calculates, it measures attenuants from transmitted light shining through particles, which is a proxy for particulate organic carbon, birefringence, cross-polarized light illuminating the particles, and that provides a proxy for particulate inorganic carbon. And then dark field allows us to actually see what color the particles are. Just to go back, Hannah's thesis was to look at particulate fluxes in the California current ecosystem. And we deployed multiple carbon flux explorers in these locations. And the interesting thing is, you've heard of the Martin curve. This is the birthplace of the Martin curve highlighted in blue. And then we've written papers about the fact that there's anomalously high flux in the wintertime in the Santa Cruz Basin. So this is the state of California. And of course, we tracked a plume of upwelled water as it went offshore. As the filament aged, if you go down the water column, different particle classes contributed to flux. And it changed depending on what location and how old the plume was. It was crazy. I'm not going to dwell on that, but we'll come back to this. But the most important thing is that they deployed particle interceptor traps down to 150 meters, and where the data overlap, we get reasonable agreement. And the Carbon Flux Explorer results show that something's going on in the coastal zone that people aren't seeing properly. That flux extrapolations are at some locations off by a factor of 7. So something is moving carbon down the water column that is not sinking material from the euphotic zone. So this is what we're doing. We've developed this thing. It's carried by a profiling float. It's outfitted with transmissometers and PIC sensor and scattering. And we now have data reduction schemes that will process the data in 7 minutes per dive and transmit the data from anywhere in the world via satellite. And we verified the codes using Hannah's thesis data. And the real question is, what is the mechanism explaining this increase of particle flux with depth? And by measuring particle size distributions and classifying this, all this is in computer codes, we can separate fecal pellet flux from aggregate flux. And when I talk about aggregates, I mean material that's larger than a millimeter in size. And what it shows is that this increase is due to the input of aggregates with depth, and we actually don't have a clue what is causing this. But that's the basis for further scientific research. But we know that these data are real because we've been able to verify them. So we're ready to go with this instrument. It could be deployed in the Southern Ocean tomorrow. But what we're going to talk about is the validation of sensors. We're going to talk about geotraces. where we compared large volume in-situ pump sampling, where samples are measured for PIC and POC, surface to bottom, 2 PIC sensors, 3 transmissometers that were deployed surface to bottom, and satellite retrievals of surface particulate inorganic carbon and POC, matched to spatially to the time of ship observations. So the most important thing, I just want to tell you where the Antarctic polar front zone is. Over here, you already know where it is, and this is the paper that we're describing. So most important thing is this birefringence sensor can detect particulate inorganic carbon. As we mentioned, this sensor is a unique inherent optical property. Nothing behaves the same way that birefringence does. This line is the base of the euphotic zone, and the line above it is the fluorescence maximum, and the white line above that is the mixed layer depths. And just to calibrate you, this is the temperature distribution of the upper 500 meters. And you can see in this PIC sensor data, a zone of relatively high PIC at the surface in the subarctic where the nutrients are measurable. When the nutrients decrease to non-measurable, that PIC concentration maximum is found at deeper depths. At the equator, there's upwelling, sufficient nutrients to support coccolithophores, and then we go down. So these data were taken in 2018. These data were done in 2022, and it's very interesting that we get a coherent overlap of the kinds of distributions that we've observed. Once you get south of 40 degrees south, nutrient concentrations build, and right here at 55 south, which is basically the polar front zone, we get the highest concentrations of PIC. And we go down here. I just want to go back quickly and just say some of the data are a little bit limited because a rogue wave hit the Ravel exactly at 55 south. And the pumps that were used to collect samples were sent to the seafloor along with the CTD and one of our PIC sensors. But we did have a second PIC sensor that we could deploy. And then this is the transmissometer measurement of particulate organic carbon showing distributions into the Antarctic. So south of the polar front zone in the true Southern Ocean at 67 south, we get very high biomasses at the surface. And the big surprise was that even though we had limited pump samples, we found a problem where there was extreme birefringence in these samples, particularly at this station 29 at 67 south. So if you flip these things on the side, here's the thermal map, thermal imagery kind of matched to the time of the cruises. And this shows that there's evidence of high PIC flux right south of the polar front zone of the subarctic front, right at the equator, and just north of the polar front zone in the Southern Ocean, which is quite cool. So we see both high export and unique distributions of PIC. So Yunhao Lee has retrieved particulate inorganic carbon. These data are actually sensor data. This is the difference between PIC concentrations from the sensor minus the sample measured. And we can see that there's a problem here. where there is PIC here, but we know from the data we don't see any PIC in the very little PIC in the samples. So something's going on here, and that's why I call it a mystery. But for most of this transect, the sensor aligns pretty well with the samples, which is pretty encouraging. The big surprise was the satellite retrievals. This is the 2-band, 3-band algorithm. That's the standard NASA product, and that shows even a higher anomaly, and also places where the satellite data are inconsistent with one another. So let's look at the profiles. The boxes at the left of this plot are the concentration profile, measured particulate inorganic carbon in the samples. And then the blue circles are the simultaneously collected data. And it shows pretty good agreement. Again, these northern stations, but again, to reinforce the fact that the sensor is quite unique. At 150 meters, there's strong variability in PIC, and we can see evidence is oceanographically consistent, and it's tied to intrusion of high oxygen water, low PIC water. You go into the Southern Ocean, and this is the problem. Station 14, we never had we lost the sample that was at 25 meters, didn't work, but the others worked, and we have good alignment. But at Station 27 in the Southern Ocean, there's really something going on where the data don't make any sense. 0 measured PIC, whereas we got measured. So there's something going on. So the question is, what could be birefringent in the Southern Ocean? And the most birefringent particle besides calcium carbonate is strontium sulfate. But it's made by Acantharia, and there are Acantharia in Southern Ocean samples. But strontium is less than 1 nanomolar, and birefringence is 10 times lower than that. Whereas we're looking to find a number like 2 to 500. Some people have talked about dinoflagellates, and then, of course, some of them are birefringent. But in the Southern Ocean, in that cold band, they're not abundant at all. So the only way that you can induce birefringence is by reflecting from surfaces. But since the sensor is highly collimated, photons impacting the sensor will bounce off and go out of the beam and never be seen in the detector. So the only way to get photons into the detector is to have forward multiple reflections, which leads the question, how does that work? And I'm going to say that chain forming diatoms are implicated, and particularly Fragilariopsis and Pseudonychia. And some people call them Brussels sprouts in class because they're apparently not very appetizing. So this is an example of an image, attenuance image of an aggregate from Phoebe Lamb's pump at 25 meters at station 14. We actually had a sample, and this is from the light attenuance measurement from transmitted light. And that's what you get when you look at cross-polarized transmitted light. You can clearly see coccoliths there, and you can see a foraminifera. There's no doubt about that. Now we go to station 25, which is one of the ones. And here's the attenuance map. And there's the cross-polarized transmitted light. There's a single birefringent particle right here. And we can zoom in on this area. So there's the attenuants. And this is boosting the exposure to actually see in more detail what's going on. And the interesting thing is that this is a group of a Fragilariopsis chain. And within that chain, there are elements that are birefringent. Some of them are highly birefringent, and we don't really understand this. So I can flip back. So we see Fragilariopsis and Pseudonychia. So we've got this problem. What causes it? This station 29, this is Pseudonychia, and it's in a colony. And this is a Fragilariopsis chain. There are other guys. This is station 25, which actually didn't have much birefringence. And this is station 14. So here's the hypothesis. Waters are weakly stratified. In this station 29, the mixed layer was 40 meters. And the hypothesis is that these chain-forming diatoms want to stay buoyant. So we hypothesize that one or more group members form a void to enhance buoyancy. And the voids would enable forward multiple reflections and a polarization signal. And thinking about this, it may be something else unique to the adaptation of living in frigid waters. And I don't know what it is, but if someone has a cold culture, I'd love to see what you see under the microscope. So I'm going to wrap up now. So 23 years ago, we deployed carbon explorers in the Southern Ocean as part of the Southern Ocean Iron Experiment. And I'm just going to talk about what happened to the float that was deployed at 66 South and stayed south of the polar front. It went through the ice edge zone. It was right in the ice edge zone. And this is day of the year in 2022. Decrease on day 11. There was a mixing event that deepened the mixed layer to 100 meters. The solar radiance corrected for the effects of clouds. And you can see at some point, basically everything is gone. And so the question is this evidence of the sinking, didn't correct that spelling, of Fragilariopsis. So to wrap things up, we want to develop the carbon observer. We know how to analyze particles and detect them and classify them. And the sensors that are designed to ride on the ocean carbon observer, we've just taken them on a 20,000 kilometer long ship track with over 100 deployments of these sensors surface to bottom. And we know where they are, and we know that there's high export near polar fronts. So we really need to deploy more of these devices and get them out in the ocean. And the Southern Ocean, of course, is a really interesting place to be. So we don't know, we can't predict the biological carbon pump and its trajectory. It moves a lot of carbon. We don't know how to assess stability without observations. And I understand that BioArgo is there. SOCOM has had a major impact on the Southern Ocean. But we think this is an additional exciting tool that can fill in the gap and provide really critically needed information. the whole issue about using satellite remote sensing of biomass to predict sinking, our experience in the California coastal regime is it doesn't work. But maybe we'll get the key for more coordinated observations. And finally, we've seen the coastal zone is underestimated as the carbon sink. So that's kind of where I want to end. And we can ask questions if there are any questions. Yeah, okay.
Great. Thank you so much, Jim. That was really interesting and quite, dense with all kinds of, really interesting information. We do have to move on to Illy, so I might ask for anyone who has a question to save till the end. Otherwise, if you pop it
Oh,
in
yeah. Oh, geez, I blew it again. You were supposed to shout out.
But if you put a question in the chat, if you wanted to monitor the chat as well, you can respond there. Otherwise, we can
Sure.
wait to the end.
Yeah, good.
Right.